Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Perkenalkan, nama saya Rio Hermantara. Saya dosen dari Program Studi Bio Medis dan Rekayasa Hayati Universitas Scientia Internasional Indonesia, atau juga dikenal sebagai HRL University. Sebagai bagian dari persyaratan sertifikasi dosen Gelombang I 2026, pada kesempatan ini saya ingin membagikan sedikit gambaran mengenai bagaimana saya mengajar di kelas genetika. Mata kuliah ini menggunakan bahasa Inggris sebagai bahasa pengantar, memiliki bobot 3 SKS, dan merupakan mata kuliah wajib bagi mahasiswa Program Studi Bio Medis dan Rekayasa Hayati serta Program Studi Bioteknologi pada semester 4. Mata kuliah genetik ini bertujuan untuk memperkenalkan konsep-konsep dasar genetika, yaitu pewarisan sifat dan variasi, serta aplikasinya kepada kesehatan manusia. Mahasiswa akan mengeksplorasi variasi genetik dalam populasi, dasar-dasar genetik, baik itu kualitatif maupun kuantitatif, serta peran genetika dalam evolusi. Mata kuliah ini juga akan memberikan wawasan mengenai bidang-bidang mutakhir seperti genetika, genomika, bioinformatika, dan bagaimana menggunakan alat-alat ini untuk memahami berbagai jenis penyakit pada manusia serta pengembangannya sebagai pengobatan personal atau personalized medicine. Adapun capaian lulusan yang dibebankan pada mata kuliah ini adalah pemahaman mendalam mengenai biologi molekuler dan seluler, yang dalam arti bahwa diharapkan mahasiswa mampu menunjukkan pengetahuan mendalam mengenai sistem biologis pada tingkat molekuler dan seluler. Dari capaian pembelajaran lulusan dan cakupan keilmuan mata kuliah, dirumuskan 4 capaian pembelajaran mata kuliah yang meliputi 1. Mampu memahami konsep-konsep yang berkaitan dengan kesehatan dan penyakit. 2. Mampu mendefinisikan dan menginterpretasikan variabel-variabel dalam lingkup kesehatan. 3. Mampu memahami kontribusi disiplin ilmu biomedis terhadap kesehatan masyarakat. Dan 4. Mampu menganalisa korelasi antara genetika, lingkungan, dan gaya hidup terhadap kesehatan dan penyakit. Dari CPMK yang telah dirumuskan, disusunlah sub CPMK yang ingin dicapai, yang diatur berdasarkan topik-topik spesifik terhadap genetika. Informasi lebih lengkapnya dapat dilihat pada lesson plan atau RPS dari genetics. Adapun materi pembelajaran yang digunakan, yaitu teaching learning materials yang dibagikan kepada mahasiswa 1 minggu sebelum pertemuan, buku referensi, beserta video dan artikel-artikel pembelajaran yang terpilih yang terdapat pada situs pembelajaran di internet. Sistem pembelajaran yang diterapkan bersifat hybrid, yang berupa campuran antara tatap muka, secara daring maupun luring, serta asinkronus, dengan pemberian materi-materi pembelajaran seperti video dan artikel terdahulu, sesuai dengan tema masing-masing sesi. Adapun bobot penilaian mencakup midterm exam sebesar 25%, group assignment dan presentation di akhir semester sebesar 25% masing-masing, dan kuis yang akan dikumpulkan menjadi 1 penilaian sebesar 25%. Video berikut akan menampilkan proses pembelajaran mata kuliah genetics pada sesi pertama. Dapat disaksikan bagaimana saya menyampaikan kontrak perkuliahan dan pemberian materi serta diskusi secara langsung oleh mahasiswa. Di akhir sesi akan diberikan kuis yang akan dilaksanakan oleh mahasiswanya secara personal. Demikian informasi singkat yang dapat saya sampaikan. Saya harap video ini dapat berguna dan bermanfaat. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Yeah, welcome everyone to this MCP genetics class, where basically if you're here, that means you have passed the MCP, so good job there. But now we're talking about genetics. So first off, welcome. And I would like to introduce the teaching team here. I'm not the only one that's going to be responsible in teaching you guys or supervising you guys. There will be teaching assistants. Let's see in the next slides. Well, first off, this is basically the CLOs. You can just read it. I will upload the lesson plan for this course if you're interested in reading these. Right, so let's just get to it. Something that's probably more interesting for you guys, which is the syllabus. So here's the syllabus for mid-semester, before mid-semester and after. We will be conducting the genetic class a little bit different than MCP because this course is designed as the asynchronous course. So 80% of every session will be conducted asynchronously, and this is through Talent LMS. Now, I should have already invited you to the Talent LMS course, the genetic course. So let me know if you haven't. References, so books. So I've listed a couple of books here, but as you can see, there's Albert's and where is it, Lourdes, which is what's more molecular cellular biology. Three major ebooks that are, well, books that I will be referring to is Brooker, Genetic Analysis and Principles, Clark, Concept of Genetics, and Pierce, Genetics, a Conceptual Approach. And all three of them, well, basically all five of them is available in the library as ebooks. Okay, well, assessments. What am I going to assess you on this genetic course? There's four things, basically something similar to MCP, right? There's the weekly quiz and assignments. Because of the nature of this asynchronous sessions, we really need to have these weekly quiz and assignments, okay? So that will weigh 30%. Midterm exam covers session 1 to 7, 25%. And then there's this final assignment and final exam. Now, these two comes together, and this is basically the genetic course project. So the project is a semester-long project. See, there's going to be a timeline because it's a semester-long, just to keep you on track. And then here are basically where the TAs will help you and supervise you. Now, the output, this is quite important. There's two outputs that I'm going to ask from you. First is a literature review. This will be graded as the final exam. And second is a popular media output. And this will be graded as the final assignment. Guidelines are provided in the file. Going into this first genetics session, which is genetics, well, introduction to genetics. And well, probably the first question that I want to ask you guys is, what do you think if you hear the word genetics? Yes, Alvin.
I think genetics is the study of the gene, genome, and DNA, sir.
Study of the gene, the genome, and the DNA. Yeah, that is entirely correct. There's the word gene in genetics, so this study might study that gene. But what else do you want to add in there? What kind of study there? Yes, Joan.
It's the aside from the ones already mentioned, it also covers the study of the inheritance of traits. Also,
traits, yeah.
Yeah, and also genetics will touch upon evolution at some point. There, either from evolution within the a population, also up to cross species. So there's this other thing called population genetics, but yes, there's inheritance genetics, and then there is also molecular genetics.
Okay, thank you, Joan. You mentioned a couple of keywords here: heredity, traits, and evolution, and also population genetics. That's part of the genetic field of study. Yeah, thank you, Joan. Those are important keywords here. Anybody else wants to add in to this discussion? What is genetics? Anybody else wants to add in here? Alvin, you want to add in?
Yes, sir. I think from what Johan also has said, sir, genetics also including the heredity, variants of the living organisms, sir.
The variants of the organisms. Yes. Yes, those are all of that is quite linked together in this genetics study. And basically that's what genetic is looking at. That's genetics, what is genetics studying, that linkage between DNA, genes, the genome, traits, and also looking at this at the molecular level and to the population level. More on that probably at the end of the part. Okay, so yeah, that's basically it. That's basically what a genetic is. But for now, I want to basically tell you a story. Hopefully can help you in understanding more into what is the field of genetics. So we first start off with probably one of the oldest question in biology is, why do we look like our parents, right? And if we want to track down this science, we can track down till whatever the age, like probably like in Roman history, or probably even older than that. But I'll probably start off with much more like a little bit modern history, where it start off with this idea, this theory. And this theory basically comes from the Greek, but was probably more refined in 1600s or so by John Baptiste Lamarck. And what this theory is called is the pangenesis. Stands for two words, pan and genesis. Genesis is the creation of an organism, as pan is everything. So what does this mean? Now, you can basically read this text. That's basically telling you that they think that, well, first, probably look at the observation, right? People usually observe first. Well, not only people, scientists observe first. They make an observation, and they probably like ask that question, that scientific question, and then they can make up the hypothesis and test it. But back then, what they see is, well, a human body is composed of like hands, head. There's the body itself. There's legs. Those are different looking organs, right? Different looking tissues as well. So they might have different informations in them. So how does the trait from a parent to offspring being transferred is that there's these informations in each body type, or not body type, each organ, and that information will be transferred later on to the sperm and the egg cell. So the zygote will have a mixture of all those informations from the hand tissues, the head tissues, and so on. So how is this being applied? One of the more popular example is the giraffe, right? The giraffe's neck, where basically there's this original short neck ancestor, where it can eat like shorter leaves, and then when the leaves are much more scarce at the lower end, the giraffe needs to stretch its neck more and more, and so on. That trait, this longer neck trait, will be passed down to its generations and generations. Now we know that that is not true, right? So this theory is a little bit flawed, but the thing is, like back then, you can't really fault them, right? Because they have limited tools to test. So that's basically why the creation of this exact tool somewhat changes or somewhat advances the field of biology, which is the microscope, right? Microscope was invented in 1600s, I think, by Robert Hooke. That's I know for sure. And basically, with the microscope, it gives the ability for human beings to see microscopic at the microscopic scale, right? So in that case, after that year, after 1600s, what they did is they proposed the cell theory, which is basically all living organisms are composed of one or more cells. And that time they can observe cells from any organisms, but probably the more popular ones are in plants, some animals as well, but also bacteria. And second is the cell is the most basic unit of life. And 3, all cells arise from pre-existing cells. Now, moving on. So now scientists know that there's cells inside, making up our human body. But the thing is, like, there's still the question, why are the offsprings same as the parents or similar to the parents? Now, there's a couple of theories. First is the pre-formation theory, which is basically that there's a miniature of a human inside either the sperm cell or the egg cell, right? Now, this theory is also incorrect, as we know it. So yeah, because of that experiment, he can reject the Lamarckian inheritance, and what he proposed is this germline theory, which stands still today, which is basically the sperm and the egg cells, well, every cells of the body carry all the information to create separate individual or organism. But the thing is, like, at that point, what they see is like there's variations, right? Well, not variations, sorry, different species. And what they think is like these species only submerged because there's that same species from the very beginning. And that is not particularly right, because what we know today, and that is thanks to Charles Darwin. So Charles Darwin back in 1840s, 40s, I think, went to the Galapagos Islands, and then he observed the habitat there. And one of the things that he observes are these finches, right? And what he observed was that these finches have different traits depending on which island there is. What's remarkable of what his observation is, he can somewhat like trace back, like these birds have these traits because the food that is available at this particular island is more appropriate for this beak, for example. And from here, he proposed the theory of evolution, which is basically like looking at these organisms have slightly different traits, right? And these slight variations would be important over time and also over changes of the environment. And from this, it's basically giving that idea that everything, all organisms might share a similar, shared ancestor, ancient ancestor. And that's where we get to Gregory Mendel. So the birth of genetics, 1865. That's basically the report that Gregory Mendel made, right? But his experiments span from 1850 something to 1864, which is basically around peas. Now, let's just ask the class here, why peas? Why green peas? Yes, Joan.
It's fairly easy to cultivate. The traits it has many traits that happen to be unlinked, as in all the traits happen to be on separate chromosomes, and they also happen to all be coded by only one gene, I think. Also, the traits are very distinct, so you can exactly judge purple or white flowers or axillary or apical flowers, or wrinkly or smooth seeds, things like that. And so, like, it's a basically very good model organism and like the very bare standards of patterns in inheritance.
Okay, thank you, Joan. That was a complete answer there. But yeah, first off, let's start off with things that Joan mentioned there, model organisms, right? So peas, and later on we'll see flies. There's bread molds, there's mice, and all that stuff. Those are model organisms. And one of the key traits, one of the key things for model organisms is that they need to be easy to grow or maintain. And also, it's easy enough to see the differences. Depending on what they really want to observe, it needs to be easy enough to be observed. And in this case, what Mendel is trying to observe is the passing of traits from parents to offspring. And that's why green peas are a really good model for that. So yeah, these Mendel experiments, we will be covering them in the next session, which is basically really important and also something that you need to understand well, not only because you're in this generic course, but also because you're a student in life sciences. Okay. Right. Thing is like about Mendel, and if you've already read a couple of things, it's like back at that time, Mendel did not get recognized, that he should have gotten, right? So his work there is like slightly undiscovered, or well, slightly as secret, right? So yeah, we will get back to Mendelian experiments later on in the next years. But probably one thing to note here is the discovery of DNA or nuclein at that time by Frederick Mischer. And so that's the first time that scientists, every level, the human populations discovered DNA, nuclein, from extracts of lymphocytes. And then in 1879, Walter Fleming was able to stain these materials. And because he was able to stain them, he was able to see mitosis, which is something that we've already covered in the MCB, right? So these are, I'm not sure, but probably, well, the text says that this is his drawing, but yeah, basically we see that the cell divisions, which is passing on the DNA from one cell to another. So this is a really good hint, right? DNA is being passed from one cell to another. Okay, so going back to Mendel, 1900s and 1900, 3 independent scientists working with 3 different plant models found that by doing what Mendel did, which is crossing over one species, well, one specific individual of this organism that has specific traits to another, will create that probability of the offsprings. That basically underlines the importance that Mendel found back in 1959. And from there, that's the birth of genetics as we all know it right now. If we ask about genetics, genetics is the study of heredity and variations. But still, like in that time, variations, what variation? What is carrying this information from one cell to another? We already had that hint from Walter Fleming, but we need confirmations, right? So that's basically what's the goal next, is unraveling that secret of inheritance. So first off, it's 1902, Walter Sutton and Theodore Boveri. Again, something similar with Walter Fleming. They observe mitosis and they somewhat like confirm on the lines that DNA is being passed from one cell to another through mitosis and also in meiosis. And then in 1911, Thomas Morgan and Alfred Studevant, 2 big names in genetics history, studied these flies. I'll ask the class again, why did he study flies? What did he use flies for his study here? Really fast reproductive. Yes, Castillo? That's correct. Alvin?
I think because like in this picture, sir, Drosophila melanogaster has a traits which is its eye color that is easily to be observed, and these traits is really related to the variants of the fruit flies itself, sir.
Okay, thank you, Alvin. So yeah, combining Alvin's answer and also Castillo's answer, which is basically something similar that what Joanne just answered the previous one, right? These models need to fulfill 2 things. It's easy enough to grow, it's fast reproductive, that's also really good plus there, and also it's easy enough to be observed. So that creates like a good model to study on. And yeah, these fruit flies are basically have those 2 basic advantages, right? So as you can see here, it's easy enough to observe the differences of a red eye and a white eye. And we've somewhat like talked about this in the MCB lab, right? Well, Thomas Morgan and Alfred Sutherland was able to create mutants by exposing them to X-rays. And basically what they did is they expose the larvae with X-rays, and then they grow them, and then they breed them, well, cross them, and then they, well, observe the traits that is being covered. And from that experiment, they found out that there was this linkage between genotype and phenotype mutations and the traits that that could be involved, that could be affected by the X-rays of the mutations. In 1941, George Beadle and Edward Tridharma. I think this was a skip back then, but this is quite fascinating. So what they did is this one gene, one enzyme hypothesis. They used bread molds, and they did something similar as Thomas Morgan. They create these mutants by exposing them with X-rays, right? As you can see at this part. And then they grow them in these media. Now, spores, these bread molds can be grown in like complete media, which is rich in nutrients, or minimal media, which is basically that basic. So probably like only glucose without any vitamins, without any amino acids. And what they get is this nutritional mutant, and that's what they observe. Now, at this point, it's something similar than Thomas Morgan, but what they did next was somewhat fascinating, which is basically they pinpoint to the mutation at hand. So this is an example. So first they have that nutrition mutant that they got from the previous picture, right? And then they grow them into like all these different tubes that contain like additional of single amino acids. And when the mold grow into a specific one, for example, this arginine one over here, that means this mold, this mutant mold, has a mutation at a gene that involves with the biosynthesis of arginine. So it's somewhat like pinpointing mutations, a gene, and an enzyme. Quite fascinating. And yeah, after that, there's basically a confirmation by 3 different experiments from Frederick Griffith, also Avery and co. on Alfred Halsey and Martha Chase, which confirms that DNA is the one that is carrying that information. Yeah, we've already gone through this in a couple of sessions in MCB, so go back and look at this. Read back your notes. And yeah, to cap that off, it's 1953, era of molecular genetics. It's basically the finding of that double helical structure of the DNA, which is something that we've covered extensively in the MCB course, right? Okay, so I've been babbling about, probably this is the last slide for this live session. Basically, again, genetics is that study of heredity and variation. Now, the thing is, like, genetics is not only humans, right? It's all living organisms. So that means the study of genetics is quite big. So when you're talking about genetics, when you're listening about genetics, it can be like quite a lot of fields. So scientists have basically made, what do you call that? Subpart these genetics into 3 things, which is transmission genetics, something like what Mendel did, right? Molecular genetics, something that you've already learned in MCB course, and population genetics, which is basically looking at the genetics, looking for the relationships of genetics and phenotype at a population level. That somewhat sums up the field of genetic research. Now, just to make sure that you understand or you gain that perspective of molecular cellular biologists, let's have a recap.